Okay, let's jump right into this explainer and absolutely demystify the Bile Esculin test. We're going to unpack exactly how this classic biochemical assay actually works, why we use it, and how it helps microbiologists solve some seriously specific diagnostic puzzles. Imagine you're peering through a microscope at a clinical sample. You spot gram-positive cocci in chains. Sounds simple enough, right? Well, here's the kicker. Visually, they all look incredibly similar. We've got a microscopic identity crisis on our hands. How do we tell apart a potentially dangerous pathogen from totally harmless, normal flora when their physical mugshots are literally identical? We can't just look at them. We have to test their metabolic behaviors. We're essentially creating a diagnostic mystery for them to solve. Here is the roadmap for our explainer. 1. The Bacterial Identification Challenge. 2. Principle of the test. 3. Preparing the agar media. 4. Step-by-step lab procedures. 5. Interpreting test results. And 6. Practical uses and limitations. Let's get started. Section 1. The Bacterial Identification Challenge, sorting the microscopic suspects. So our primary goal here is highly targeted. We're trying to isolate and identify Enterococci and Group D Streptococci, like Streptococcus bovis, and definitively differentiate them from the non-Group D viridans group Streptococci. This is an absolutely crucial distinction in a clinical setting, mainly because these organisms have vastly different implications for patient care and, crucially, antibiotic resistance. Misidentifying these suspects could easily lead to the wrong treatments, so we really need a specialized tool to draw a hard line between them. Section 2. Principle of the test. The Bacterial Stress Test. To set up this little stress test, we use a notoriously harsh chemical environment to weed out the weak bacteria and see exactly what the survivors can digest. Enter esculin, the namesake of our test. Esculin is a glycosidic coumarin derivative. Now, simply put, it's a molecule made of two parts, a sugar called glucose and a compound called hydroxycoumarin. These are linked together by an oxygen ester bond. For a bacterium to actually use this as a food source, it needs the specific molecular scissors to cut that exact bond. And here's the first step of that molecular chain reaction. If a bacterium possesses a specific enzyme called esculinase, it can just slice right through that ester bond. This hydrolysis reaction breaks the esculin down into beta-D-glucose, which the bugs happily consume for energy, and a leftover byproduct called esculetin. Now, this brilliantly illustrates the dramatic visual climax of the chemistry. That leftover byproduct, esculetin, is highly reactive. When it leaks into the surrounding agar, it acts like a magnet for iron. It binds right to the ferric ions, the Fe3+, that we've intentionally seeded into the media. When these two meet, boom, they form a phenolic iron complex that turns a dark brown or jet black color. That color change, that is the definitive chemical footprint we're hunting for. Section 3. Preparing the agar media. Setting the chemical trap. You know, you can't just toss bacteria and esculin into a tube and expect a clean result. You really have to set up a series of chemical booby traps. Here are the key ingredients that make up the bile esculin agar. Obviously, we have our esculin, but the absolute most critical component is the ox bile. We're talking a brutal concentration of 40% bile or 4% bile salts. It acts as a massive inhibitor, stopping almost all other gram-positive bacteria dead in their tracks. Only the absolute hardiest bugs survive this. Then we add ferric citrate acting as our indicator by supplying those iron ions. Finally, beef extract and peptone provide basal nutrients, and agar solidifies the whole shebang. It is a perfectly engineered, selective, and differential environment. Section 4. Step-by-step lab procedures running the test. Depending on the speed and resources you have, there are two main ways to run this test. First up is the traditional tube test. You take a well-isolated colony from an 18 to 24-hour culture using a sterile loop or needle, and you inoculate the surface of the agar slant. A quick tip here, make sure to leave the cap slightly loosened to ensure adequate aeration. Then you incubate the tube aerobically right at body temperature, so 35 to 37 degrees Celsius. The catch here is the waiting game. You typically need to observe it at 24 hours and sometimes up to 48 hours to really confirm that color change. Alternatively, we have the rapid disc test. Talk about a massive time saver. You take a commercially prepared esculin disc and moisten it with just a single drop of distilled water. Literally just one drop. You really don't want to saturate it. Then you smear 2 or 3 isolated colonies directly onto the disc. Instead of waiting days, you just leave it at room temperature and watch it for about 10 minutes. If the bacteria are packing that enzyme, the disc will turn dark brown or black almost right in front of your eyes. Section 5. Interpreting test results. decoding the clues. Now, a test is really only as good as the clinician interpreting it. Let's compare the actual visual outcomes. A definitively positive result requires two things: gross on the agar, proving the organism survived the bile, and a distinct blackening of the medium, proving esculin hydrolysis. A negative result is characterized by a lack of color change. But here's a huge caveat. An organism might grow luxuriantly on the agar because it tolerates bile. But if the agar doesn't turn black, the test is negative. Growth alone absolutely does not equal a positive test. Or obviously, the bile might just inhibit the bacteria entirely, giving you zero growth and a negative test. Here's a surprisingly cool hidden clue you can use, especially if you happen to be working with a broth that lacks the iron indicator. Esculin is naturally fluorescent under long-wave 366 nanometer UV light. If the bacteria successfully hydrolyze the esculin, that fluorescence gets completely destroyed. So a positive test actually results in a loss of fluorescence, while a negative tube will just keep right on glowing under the black light. Let's see how our specific suspects react under these exact conditions. Enterococcus faecalis thrives and turns the agar black, classic positive. But look at Escherichia coli, a gram-negative rod. It grows perfectly fine because it isn't inhibited by the bile, but it lacks the esculinase enzyme under normal conditions, so it remains negative. Meanwhile, another gram-negative, Yersinia enterocollitica, both grows well and turns the agar black. This just goes to show exactly why understanding the precise scope of your test is absolutely vital. Section 6. Practical uses and limitations. Caveats of the case. The primary practical use of this test is the rapid presumptive identification of Enterococci and the Streptococcus bovis group right out of clinical samples. It cleanly separates them from other viridans Streptococci. Beyond that, the selective nature of the bile esculin agar makes it surprisingly handy for isolating a few other specific pathogens, namely Listeria monocytogenes and Yersinia enterocollitica, since they also have the ability to tolerate bile and hydrolyze esculin. We do, however, need to be hyper-aware of false clues. If you apply way too heavy of an inoculum to your slant, you can actually overwhelm that 40% bile concentration. This allows weaker bacteria to grow and potentially trigger a false positive. Also, bacteria that naturally produce hydrogen sulfide during their metabolism will react with the iron to create a black complex, totally mimicking a positive esculin result. Even our earlier suspect E. coli, which actually has a beta-glucosidase enzyme, might slowly turn the test positive if you let it incubate for too many days. And this brings us to a final, absolutely vital takeaway to consider. Why must the Bile Esculin test always be just one piece of a larger biochemical puzzle? Well, because biology is rarely absolute. While this test is an incredibly powerful tool for filtering out non-Group D Streptococci, its limitations, like those false clues from hydrogen sulfide or super heavy inoculums, mean it can never be the sole judge and jury. It's a fantastic presumptive indicator, but to truly nail down a microscopic suspect, it always has to be combined with a wider array of biochemical tests. I hope this explainer illuminated the brilliant chemistry behind the Bile Esculin test for you. Keep questioning, keep investigating, and I'll see you in the next explainer.